All right, good morning, everyone. Seems like we lost a few people, but perhaps that's one more. I hope you all got started a bit with quantitative proteomics yesterday and had chance to work your way through these tutorials to some degree. As I said, if you're running a bit late, that doesn't matter. Just talk to us and we will help you getting through them if you get stuck. This morning, I would like to talk about quantitative proteomics. That's basically the second half of what we tend to do in proteomics. This is not about knowing what is in there, but knowing how much of something is there. So what we're going to do is we will talk about quantification using mass spectrometry. And I will start again by defining some basic terms from analytical chemistry. What do we mean when we say quantify? Everyone seems to have some idea of what quantification means, but there are distinct differences when you talk to an analytical chemist versus to a biologist versus to a statistician. And I'll try to sort that out a bit, and we'll see how far we get with that. And then I will talk about one strategy for quantification. There are others. I will talk about label-free quantification. You might know that there are other approaches like SILEC or MRM or PRM or SWOT. Or I think the list is almost endless. There must be more quantification techniques than mass spectrometrists around. But let's start with basic analytical chemistry. So who in the room ever had a course in analytical chemistry? That's great. Then you can check whether that's in agreement with what I'm going to tell you. For the other ones, analytical chemistry is actually something rather straightforward. We have been doing it yesterday without me telling you that we were doing analytical chemistry, because everything we have been discussing the first part of the lecture, separation techniques, that's all analytical chemistry at heart. So analytical chemistry is a study of separation, identification, and quantification of chemical components. And of course, today we're dealing with this quantification part, and we will see what that really means. If you look that definition up, then a quantification is defined as the act of counting, measuring that maps human sense, observation, and experiences into members of some set of numbers. That's probably as broad as you can get. It basically means we're trying to attach numbers to things that we can see or observe or measure. Now, we are dealing here with quantitative mass spectrometry. So we will actually use a mass spectrometer to turn amounts of analytes into numbers. That's all we mean by quantification right now. Note that I'm just saying numbers. I'm not talking about units. I'm not talking about counts. I'm just saying numbers. So let's start with some terms. When I'm talking about an analyte here, then I will be talking about the stuff that we want to measure. In our case, proteins, or later today, metabolites. So we're talking about molecular substances, and this is usually embedded when we're doing biology in a matrix. And a matrix can be anything else. It's everything that the analytes are hidden in. If you're looking for proteins, that might be all the other debris that comes from a cell. Lipids, water, salts, whatever you have in the cell. This matrix is usually not helpful when you're doing the analysis. And we will see what it actually causes. It can significantly impact the way the whole analysis needs to be performed, because you might have to separate it. And then we are back at the part we discussed yesterday. How do you separate things? So an example might be you do proteome analysis from urine. Urine contains proteins, peptides. That's what we're interested in, the analytes. And all the other stuff, that is matrix. Water, metabolites, urea, we don't need that, and we need to get rid of it. If you want to quantify analytes, if you try to attach numbers to it, then these analytes tend to be in solution. We usually don't do proteomics on solid substances. We usually inject a solution of our analytes. So we're actually not dealing with absolute masses. We're not trying to weigh the thing, but we are trying to determine the concentration. And the concentration is the amount of substance per volume. And that can be measured either as a molar concentration, C, which is then the molar amount per volume, or it could be a mass concentration, in which case it's just a mass per volume, always of the analyte. Translating molar concentrations into mass concentration can be done via the molecular weight, but you know that already. That's really chemistry 101, rather straightforward. Whenever we measure something, there are ways in which this measurement can go wrong. And that is usually referred to as an error. And this error comes in, let's say, two basic flavors. I try to illustrate that with here this target. If our true value is somewhere here in the middle, and you do 10 repeated measurements of your same quantity, you would expect them to be not always exactly in the bullseye. That would be too good to be true, but they scatter around that. And there are different ways in which they can deviate from this bullseye. In the left-hand case, you see that they're scattered more or less symmetrical around the bullseye. And that is what you call a good accuracy, because if you average over it, it captures the true value reasonably well, but poor precision, because repeat measurements will actually deviate from another. On the right-hand side, you have another case, a bit extreme. Here you have a good precision, which means that the variance between measurements is comparatively small, but the true value is off. So you have a bias, a systematic error in your measurement, and both types of errors occur. In an ideal experiment, you would combine high accuracy with high precision. When we think of these two types of errors, then sort of the intuitive way of thinking of this precision is as a random error. This random error has usually or often normal distribution. So if you have enough repeat measurements, you can get closer to your true value. Whereas a systematic error or bias, you can repeat the measurement for as long as you like. You will not get further to your true value. So there are some caveats with that. Now, if we want to determine and analyze what you learned in your analytical chemistry course, is the standard approach for that is usually do a calibration curve. So you take a stock solution, you pipette that in 15 concentrations, do 15 injections, and what you note down is the detector response. This could be something like the peak area or the peak height, or it could be just a number that your detector returns. And you plot that as the stock concentration that you actually injected. And what you should expect and observe is that there is a linear response of your detector. It doesn't have to be the case, but in mass spectrometry, as we will see, it's actually a decent approximation. But at some point, with every technique, you will observe some sort of saturation. So why do we observe this saturation in a mass spectrometer? Why can't the mass spectrometer go on for all eternity?
Either detector response or ion source related issues.
Yes, but what? Those are the two. Yeah, it's the ion source. It can be the ion source. It can be the mass analyzer or the detector. These are the three components. All three of them can contribute that. But in which way? Can you imagine what are issues with that? Well, what would it mean if there's no saturation? It would just mean that we can get arbitrary signal intensities. So we could argue we would also be able to measure a concentration of 1 ton per milliliter. But obviously, we simply can't have that because, first of all, we can't put that concentration in solution. Second of all, we tend to have saturation effects in the instrument. The trap has only a limited capacity, and once the trap is full, you can put as much stuff into your ion source as you like. You will not get more ions into this trap, and accordingly, the detector signal will level off at this maximum capacity of the trap. And there are similar issues also when it comes to the detector. So what is really interesting to us is how far can we go? So how far up can we go until this linear regime ends and the detector starts doing funny stuff, or the instrument? And that is typically known as the linear range. There are different quantities that we can determine from such a calibration curve. First of all, we have the LOL, which is the limit of linearity. We have the LOQ and the LOD, the limit of detection and the limit of quantification. These are the concentrations where we can, for the first time, really see our analyte in the signal. And this is a level where we can quantify it reliably. And we will see what that means in a second. And you would hope that if you are in this linear range, meaning beyond the concentration where you can reliably quantify and below the concentration where your linearity goes away, then you can actually quantify with a very simple linear calibration. And the slope is the sensitivity. Now, not all detectors have a linear response, but for mass spectrometers, that tends to be true. And we like that because it makes many things a lot easier. You could argue you can also have nonlinear calibration curves and you can do all sorts of funny stuff. But in analytical chemistry, that's usually as far as we go. So why is there a level of detection where we can see the analyte, and why is that distinct from the level of quantification? What does it mean if we see an analyte?
Basically, we get close to the noise, so we can still maybe the noise detect it, but be able to quantify it against would be not very reliable since we're not sure if it's just noise we're quantifying or if it's actually the detect what we wanted to detect.
Yeah. The noise is here the limiting factor. If you go down to the level of the noise, then the analyte signal becomes indistinguishable from a noise signal. So you have to be above the noise. And this level of quantification actually implies that you have some sort of confidence. So you need to specify at which confidence you want to quantify that, and then you can determine this level of quantification. This detection limit, as has been pointed out, is related to this noise level. Basically, you can imagine that as if you are not far enough away from the noise level, you will not be able to reliably pick that up. And for the limit of quantification, you need to know whether you can distinguish two values with reasonable confidence. And if you specify that, you also know how far down you can go. The problem, what's the problem with this type of calibration? Why does barely anybody in proteomics do calibration curves on their instruments? Say, yes. If you want to quantify 10,000 proteins, what do you need to do? Well, you need to isolate 10,000 proteins, determine their concentration in a stock solution, dilute them down, inject them, run them, do the calibration curve. It's not a recommended way to try and get your PhD. This is not going to work. It just takes a lot of time, and that is why we tend to go another route. So let's look a bit more at this quantification with a mass spectrometer. So when we look into a mass spectrometer, then that two processes here that we need to consider. One is the ionization. It's not obvious that the number of ions that goes into mass spectrometer is really proportional to the concentration. And as I said, there are also effects here that lead to nonlinearities. But you can imagine that if there are more ions in your analyte in the spray, then the odds of that being ionized also increase. Up to a certain point, if you have too many matrix or analytes in there, then not all of them might be ionized. So there's saturation and there are interference effects here as well. But let's assume we have a source that really gives us a number of ions at the entrance of the mass spectrometer that is proportional to the concentration in the analyte. Then you can still have these saturation effects here in the trap part. And you can also have saturation effects in the analyzers. What is shown here is an Orbitrap analyzer. Who has ever seen an Orbitrap? Who has ever held one in their hand? They're not that huge. They're comparatively tiny, and you can imagine that there's a limited capacity to them. What's really the problem with this capacity? Well, the problem is that these ions bear charges. So if you try to pack them too tightly, what happens? These ions repel each other. So you can't pack these ions as tightly as you could pack the analyte in water. So there are some effects here that come from charge-charge repulsion. So if we want to do LC-MS on a quantitative level, then there's one problem here. And that is our analyte will elute over time. So we will actually not see, unless you do direct infusion mass spectrometry, we will not see a signal that is directly proportional to the concentration, but we will see a signal that is proportional to the concentration over time as your analyte elutes. Now we're making an assumption here. The first assumption, or several assumptions, the first assumption is that we inject the fixed volume. So when you do 10 measurements, you always inject the same volume. That's helpful. Otherwise, you would need to correct for the volume. It's not a big deal, but it's annoying. We also assume that this column doesn't lose anything. So whatever we put into the column will eventually come out of the column, which is a brave assumption, but for simplicity's sake, let's just say it's true. If that is true, then the analyte can spread out over time, but everything that goes in front of the LC column has to come out at the end at some point. So we can argue the overall amount of analyte is the integral over the total complete elution time. If nothing gets lost, then if you integrate over time, you will get everything out. Why is comparing the height of these peaks not a good idea? Yes. So if things elute at different times, then the width of these peaks can change. Which peaks are broader, early or late? The late peaks have more time to diffuse within the column, so they will be broader. And things can shift in retention time, as you probably have all seen, and that means that also the intensity goes down. The area under this curve, the integral over that, has to remain the same. But the intensity, the height of the peak will actually change depending on the retention time. So it's a good guess to assume that the area under the curve is a more reliable, more robust estimate. The problem when you compare analytes is, however, that this ionization efficiency differs. So there is no absolute number attached to that. If you start with 10 micromole per liter of one compound and compare the peak intensity to 10 micromole per liter of a different compound, then the areas under the peak will be vastly different. That can be many orders of magnitude in difference. Where does that come from? It comes from the different ionization efficiencies. One molecule will be ionized with 90% efficiency, the other one with just 1% efficiency, and accordingly, you get fewer ions that actually enter the mass spectrometer. So if we try to formalize that a bit, we could say if the MS signal intensity is proportional to the concentration eluting off the column, then this concentration is, or this factor in this proportionality here, that's our sensitivity for this particular analyte. And what we record is this intensity as a function of time for the analyte I, which means we're looking at one particular mass. Different analytes come with different masses. And we can just say the intensity as a function of time is this proportionality factor times the concentration at the end of the column. This is not the stock concentration. This is the concentration as it elutes off the column. If we want to know what was really in our stock solution, then we need to integrate all the signals. So the integral over the signal intensity is the integral over the concentration times the sensitivity factor. We can just pull that out. It's proportional. These elution profiles tend to be roughly Gaussian, and roughly means they can also be very non-Gaussian. But the shape does not violate this integral condition. Yeah? No matter which shape, whether it tails or heads, it does not matter. It all comes off the column. So we could argue if we can model this concentration profile here as a Gaussian, then we basically take the overall concentration, the total concentration in our original sample, we multiply that with a Gaussian function that is centered around the retention time of the analyte, the median retention time, and some peak width. And that basically tells us what the concentration would be over time. And now if we integrate that, if we put that into the equation from earlier, the integral over the intensities is F times, well, the total concentration we pull out of that, and now the integral of this Gaussian, and that is, of course, 1. So what that really shows is that the signal intensity, integral over our signal intensity, is just the sensitivity here times the total concentration. And that is the basis for quantification with mass spectrometry. It just tells us we have a linear relationship between the integral of the signal and the total concentration in our injected volume. When we look at which parts of the proteome we can detect, identify, and quantify, then we also see a few differences. More peptides and proteins are usually identified than are quantified. Why is that the case? Shouldn't you be able to quantify everything that you see? Yeah. If we say quantify, it's not sufficient that we just see it. However, in order to trigger, by now mass spectrometers are so fast that in order to trigger a tandem spectrum, which we need for the identification, oftentimes you need to be barely above the noise. So you can still get a tandem spectrum, even though you don't have enough of an elution profile to actually quantify the analyte. It depends a bit on your sample. It depends on the quality of your tandem spectra on your instrument. But roughly what happens is that you can detect a lot, you can identify a bit less, and then you can accurately quantify a subset of that. In metabolomics, things are slightly different, as we will see this afternoon and tomorrow morning. Here, identification is much harder than in proteomics, so we tend to identify a lot less than we can quantify. Actually, several orders of magnitude less than we can quantify. And that's one of the challenges that we have, just as a forward pointer to what we will discuss later today. Now, In order to deal with some of this variation that we have with the quantification, people have come up with different strategies for quantification. And the most popular way of dealing with that is actually by labeling. Labeling, you all know from lab work, it's just a way of attaching things to other things, to analytes, for example, fluorescent dyes to things that they light up, GFP. It's a way of labeling the protein that you're interested in. Now, since our mass spectrometer can really only see mass over charge, the only way we can label things for a mass spectrometer is to change the mass. And the most convenient way to do that is actually using isotopes, stable isotopes of the same element that are basically chemically the same, but behave different in the mass spectrometer. So if you introduce a label in one sample and no label in the other, then you can keep the same analyte in two different samples apart, because one of them will be shifted. The advantage is that if you can now reduce the variance because you treat the two samples identical. That makes it very easy to annotate them, as we will see in a minute. We just look at the spectrum. We have two peaks, and we can see, well, they're basically same size. There's probably going to be the same amount of that. But labeling can also has some disadvantages. It can be expensive, it can be difficult, it can be unreliable to introduce. You need to introduce it quantitatively. And labeling is not always possible. In particular with clinical samples, there are some labeling techniques that would not work. Metabolic labeling, you would not be able to feed the patient isotope labeled amino acids, and you would not be permitted to. Also, it would be expensive. You can label mice, but humans, Silac human, nobody has done that as far as I can tell. It would be very expensive. So there are fundamentally two chemical ways to introduce these labels. One is called chemical labeling, where the peptides are modified chemically after extraction, and that can be done with clinical samples as well. The label must be attached covalently to a specific functional group, and that can be the N-terminus or specific amino acid side chains. It does not involve a perturbation of the in vivo system, which is quite helpful in certain contexts. But the labeling occurs late during sample preparation and thus does not account for all the variation in the early steps of pre-analytics. The other option is metabolic labeling, where stable isotopes are integrated by feeding the organism with labeled metabolites, either amino acids or nitrogen sources or glucose. And as you can imagine, that means that all the proteins need to be turned over. So it takes a while until every amino acid and every protein in cell culture has been replaced by the stable isotope variant. Also, it introduces a perturbation to the in vivo system, because while these isotopes are chemically the same, they are not exactly the same. Heavy water is not the same as light water. The boiling point, for example, of heavy water is not at 100 degrees Celsius, it's at 105. So there are subtle differences between these isotope-labeled compounds. The advantage, however, is that the labeling occurs early in the study, and that can result in higher reproducibility, as all steps are actually covered from the generation of the data. What we will discuss here is another way, that is label-free quantification, that to me, at least, is the most natural way of quantifying. You don't introduce a label. You remove further sources of error, because you don't have to do the derivatization of the peptides. But the data needs to be acquired on different runs. That causes some variation. And in order to analyze the data sets, you need to have some software support, because you can no longer inspect individual spectra and just measure the height of the peaks. Instead, you actually need to compare across runs, and the data sets tend to be rather extensive. This figure gives you a rough idea a rough overview of the different labeling techniques. Metabolic labeling would start here early on in the cells or tissue, and then would introduce the two labels, blue and yellow here, throughout all steps of purification, extraction, digestion, data analysis. Whereas chemical labeling would start later on, because you would only introduce it at the level of the peptides usually. So whatever happens here, you would not capture in those. And label-free basically does nothing to the sample, and that means you will have to figure out what happened in between using statistical means. And that is why this data analysis becomes a bit harder. All right, any questions on these basic analytical chemistry terms? Yes, please.
Specifically in NMR metabolomics, we use some internal standard against which we quantify the metabolites, whether we are targeting some specific ones or not targeting. So in this kind of proteomics analysis or in the analysis, we have such kind of internal standard.
Yeah, you can do that as well. You spike in a peptide and you measure relative to this peptide. It's, you can use it, but it's rarely used. What do you think? So first of all, your peptide is only going to tell you the relative concentration of the same peptide. They're usually labeled, and isotope labeled peptides are going to be expensive. And in my experience, if you spike in just 2 or 3 peptides and try to normalize with those, the error that you introduce is much higher than if you use other ways of normalizing the data. I know that people use it in an NMR. In metabolomics, every single experiment that I've seen where people used up to 10 or 12 peptide, not peptides, metabolites, it depends on what you actually want to see. In the end, we just discarded these internal standards. So it's rarely done these days. Okay, a few more terms now, more related to label-free quantification. Yes, please. iTrack and TMT? Yes. Where would they fit on this chart? Well, they would actually be chemical labeling. They would be introduced at a level of peptides. So it's a covalent modification of the peptides that is introduced after a digest. Okay, but let's forget about all this label crap. Let's just get a clean slate, start with just proteins, and try to quantify the heck out of them. So when you look at the data that we deal with, you just take your peptides, inject them, LC-MS run. What does the data look like? You've seen some of the data yesterday. Fundamentally, our mass spectrometer only measures spectra. So our data is always going to be mass spectrum after mass spectrum. You got 50,000 mass spectra. Forget about the tandem mass spectra. We don't care about those anymore. Those were the stuff of yesterday. Today, we're just talking about MS1 mass spectra. And what you would see is if this is a time axis, one spectrum after the other, you would see stuff coming up, these peaks here, stuff going down, stuff coming up. This might be a next peptide eluting from the column. It's a simplified representation. This is just a very tiny bit of one of these measurements, as you can imagine. If we take these spectra and sort of have a bird's eye view on top of them and just color code each peak by intensity, then this is what it looks like. So now you have spectra going this way. Blue or dark is high intensity, yellow is low intensity, and this is retention time. So what do we see? Well, we see as time goes on, we see stuff coming up, intensity going up at one certain mass and then going down. This is a different mass, and we see the isotope profile. So we actually see adjacent monoisotopic plus 1 plus 2. Monoisotopic plus 1 plus 2 plus 3. And this here is a 3D view where you see it even more clearly. Retention time, and you see stuff going up, almost Gaussian illusion profiles, the way you would like it. And what we would need to do now, when you think back what I just showed you, is integrate all the signal that belongs to one analyte. So people have called that many things in literature. Metabolomics people tend to talk about peaks and mean chromatographic peaks. People have used the term 3D peaks for everything that belongs together. I will try to define these terms. We have been using them like that for a long time. I'm not going to change them. Whether you like them or not, that's up to you. Either translate it to what you have in your mind, to your internal vocabulary, or accept it as it is. I would use two terms. One is the map. And a map is just this two-dimensional data set that contains all the signals from one LC-MS run. So that's what you see in the background. Again, where's retention time? Well, retention time is going this way. This is retention time. This is M over Z. And you see stuff coming up, stuff going down. And you see they appear not as individual peaks, but actually as sets of peaks that are spaced at fixed intervals. Well, what is the distance between two of those? It's roughly 1 Dalton over the charge. So if it's a charge 2 peptide, then they're 0.5 mass units apart. And all that signal that comes from one analyte, we call a feature. Or to be more specific, all the signal caused by the same analyte in a specific charge state. Because some of these peptides occur in multiple charge states. So technically, you see two features from the same peptide. You see a +2 and a +3 feature from the same peptide, but the overall concentration would be the sum of the two. And the process of finding these guys, that is what we call feature finding, or people use also feature detection. That means find me all the peaks in the original mass spectra that really belong to my analyte. So this here is a very tiny, I guess, 1/10,000th of one such map, roughly. And when you look at this, you see noise, all this grass here, as we like to call it. The grass is growing on this map, and it's hiding some of the signal. That's why it's not that easy to detect what is really what here. But clearly, the high-intensity ones are easy to pick out. So when you look at these maps, you can easily spot some of these features. And what we would now like to do, and that's where we need algorithms for, that is figuring out that all these peaks here are the same feature. They come from the same analyte. So we would like to know how much is there. We want to quantify that, and ideally, we would also be able to identify that. But we know how to do that. If we have a tandem spectrum, we attach the tandem spectrum to that. And then we do protein inference, and you can figure out the rest from there. In label-free quantification now, once we have these, are able to find these features, the overall strategy looks roughly like this. The first step is always we find all features in all maps. So in this case, we have 3 samples, blue, green, and red, and we try to quantify all the proteins in these 3 samples. So we find these features. What does that mean? Well, we turn these maps that contains millions or hundreds of millions of peaks into things that are more discrete and more interpretable. So all the signal that comes from this part of the map here, we turn into one feature, illustrated as a dot. What are the properties of a feature? Well, we know the retention time, we know the mass over charge, we know the charge, and we know some sort of intensity. We know the integral over all the signal, because that is what's going to be proportional to our concentration in the sample. What we also need to do is we need to align them, because chromatography is not very stable. It tends to shift. So we need to correct for that by aligning these maps and figuring out that this guy here is still this guy, is this guy in the other map, even though you see in the original map they were slightly shifted with respect to each other. Yes, please.
So when you say integral over, so it's the integral over the sigma of the same analyte, right? Or
Yeah. Everything that belongs to one analyte, and I'll explain how we find that or not in a bit. But basically, every signal caused by an ion from this analyte. Yeah, and they can be spread out over retention time because they elute at different times, and they can be spread out over m over z because some of these ions will be monoisotopic, some will have higher isotope profiles. And the next step is then to link these guys, so to really figure out that this dot here is this dot after the retention time alignment, and then attach an identification to that and a quantification, because already if we have correctly quantified these guys here, then the intensities should already tell us the relative quantities across these maps. So we would figure out that it is roughly half the concentration in the red map as it is in the other two maps. Yes, please.
Would alignment of the maps fail when you have PTMs?
No, because you would see the PTMs as distinct features. It depends a bit on the PTM that you have, but for phosphorylation, for example, it uses a different charge and a different mass. So it would shift the feature in mass by 98 Dalton. And it tends to shift it in retention time as well. Why is that? Because it introduces an additional charge. It would also change the charge state, by the way, because it reduces it by 1. So you tend to have them as very distinct features. Depending on the modification, some modifications are, of course, less prone to shifts. As a matter of fact, when you look at this data set, I haven't really talked about that, but what you see here is actually a labeled data set, because when you look closely, you see pairs of these guys. These are very subtle modifications that differ only by 4 Dalton, and they have a small shift in retention time and just shift by 4 Dalton to the right. But you would find them as individual features because they are individual analytes, a labeled and unlabeled version. Okay, so that's what we want to do. How? Yes, please. It seems so, yeah. The more interesting question is, if you have 3 tandem spectra attached to these 3 guys and they all say something differently, that's when it gets interesting. We do that occasionally, works as well, but these identifications, particularly when since we're using DDA, tend to be a bit erratic, in particular for the low intensity features. So for the high intensity features, it doesn't really matter which way round you do it. But for low intensity features, it can actually make a difference. And there is a balance between doing good identification and good quantification. Again, you can't have both at the same time. What happens if you measure tandem spectra? What happens to your elution profile? Let's see whether I got a slide for that. You can't see it here. But what happens if you measured tandem spectra? Well, it's going to slow down your MS1 cycle. So if you get more identifications, you get a poorer quantification because you're losing temporal resolution. So rather than like having 10 points along the whole retention time, you tend to have only 5 because the mass spec is busy measuring all these tandem spectra. So doing top 40 is not a good idea if you want to do label-free quantification. And this is where it can actually help to link identifications across maps, because you can measure some of them in one sample, some in the other sample, and this way link the evidence across them. Yes, please.
So, if you have, like, let's say, 3 samples, and you see a peptide that is more abundant as a +3 charge state, and then on the other one, maybe +4 charge state, how does the strategy overcome that in terms of quantifying?
Let's see how we do it first, and then we will discuss these issues. Yeah, because that is clearly one of the issues. All right, so that's the rough outline. So how do we get there? So this whole feature finding process can be seen as a data reduction process. Now you start with your sample, do the HPLC-MS, and you get raw data. This is an old data set. You start with like 10 gigabytes of data, bunch of samples, and you reduce that by doing peak picking and by reducing it to maps, by feature finding, and then you annotate them with the identification, and you ask only which are differentially expressed, and then you end up with roughly a kilobyte or a bunch of couple hundred kilobytes, depending on how much is differential. And fundamentally, you're just boiling down this data step by step. This here is centroiding, this here is feature finding, this here is a statistical analysis. And in this way, you break it down to the stuff that's really meaningful. So this whole analysis that I'm describing is a way of extracting what's essential, what's relevant out of a huge haystack. And finding this needle, these 10 proteins that you really care about, this is what makes label-free quantification interesting. So let's look at feature finding. Yes, please.
So in that feature matrix for the statistical analysis, when we have that data, do you also consider interactions among proteins or peptides in the feature, or interaction among features?
What do you mean by interaction of features?
So in this raw data, when we have that Excel sheet, not every location.
We don't have an Excel sheet yet, and we don't want to get one. Yeah, but let me try to figure out how we get to such a table, as you call it, and then we can see what we do with that. So how does this work? This is a small excerpt from one of the maps, and when you look at that visually, you would say, well, there's a feature here. Clearly, this here is the most intense part. We can look at retention time, mass to charge ratio visually by doing that. Of course, you can't do that for 150,000 features. So we need to do that automatically. And ideally, we would like to learn a mathematical model that describes the feature intensities. As you can see, this is low resolution data. Things are a bit more resolved these days. So let's think about what we want to extract from that. So if we see these roughly 1,000 peaks or 2,000 peaks that form the whole data set in this region, we want to figure out which peak in which spectrum is in. This guy here is probably in the grass. This is out. This most intense guy is clearly in. But where do we draw the line? Where do we say this peak is in, this peak is out? That's sort of the problem with this integration. It's not that straightforward because it's spread out over 2 dimensions and you need to find all the signal. If we make that decision, what would we want to extract in terms of information? So what's the output of our algorithm? Well, the output would be ideally a model of what should be included, and that has certain attributes. And one attribute is clearly the position, so mass to charge ratio and retention time. We need that in order to link it back later on and also link it back to the identification, because that is done via the connection of the MS1, MS2 spectrum. We want to have the intensity, which would be roughly the volume under this model. That is the integration. That's what we are interested in. And we would like to have some idea of the quality. So does that really look like a Gaussian peak, or has that a very odd shape? So most of these feature finding algorithms assume a feature model, some statistical description of the signal that we have. And it's fairly straightforward because we know the physical processes that lead to this feature. We know that there is a separation process along the retention time that should result in a Gaussian elution profile. We know that there is a separation with respect to mass to charge ratio, which should lead to a bunch of peaks that adhere to binomial distribution. So if you just multiply these two models, because these processes are entirely independent, then we should get a two-dimensional model of what to expect. Let's start with this model for the mass spectrometry. I said this is a binomial distribution, but there's one part missing. We simply don't know which peptide. Now you could say I use the identification data, but if you haven't identified this thing, then you're in trouble. So that would mean you can only identify stuff that quantify stuff that you have identified. People came up with a trick for that a long time ago. They basically said the composition of proteins is roughly the same, independent of their sequence length. And that's where the notion of an averagine comes from. This averagine is roughly an average amino acids. So you take the 20 amino acids, look at the sum formula of each amino acid, and average that. Now you could argue that is a very rough approximation. Glycine is clearly different in composition than arginine. It has different number of atoms. But since we're looking at peptides that are at least 10 amino acids long, this averagine is actually a very reasonable assumption. So you can average that and you get 4.94 carbon atoms, 7.76 hydrogen atoms, 1.36 nitrogen atoms. That doesn't mean that we suddenly have split atoms. It just means that in order to estimate what that distribution should like at a given mass, you can just divide this mass by this averagine, and you get something like it should have on the order of 101 carbon atoms. Whether it's 101 or 105, in the grand scheme of things, that does not matter. This distribution will not look very differently. So this rough estimate gives you a good idea of what the isotope distribution should be at a certain mass. And this is how we then can compute it. I've showed this slide yesterday already. When you have this averagine approximation, you can easily figure out at a given mass, should it look like this or should it be like that? But it means when you're doing feature finding in different regions of the map, you will need to look for different patterns. You will need to look for different models. The model is always going to be mass dependent and charge dependent, of course. The second thing is that different instruments have different resolution. That plays a role for low resolution instruments, but surprisingly, I thought this problem had gone away. And then I started doing top-down mass spectrometry, and we ran into the same problem again. If you have charges of 50, then the separated peaks are no longer separated. So basically, you can fold your model with a rough idea of what your peak width is going to be. And this is the mass spectrometric peak width. That's the resolution of your instrument. And on a high resolution instrument, you will have this isotope pattern nicely resolved. And as you go to lower resolution instrument, you can see that you have no longer baseline separation, or you get this smoothed out curve. It's just one parameter in the model. Once you know what your resolution is, you know what to expect in these mass spectra. On high resolution Orbitrap TOF instruments, for peptides, you will always have base peak separation at this point for all reasonable charges, except if you do very large peptides, top-down native. When you bring that together with the elution profile that tends to be Gaussian, then there are different caveats there. There are a bunch of papers that describe different elution shape profiles. I think there's one that has 93 different equations for elution profiles. You can have your pick. Ultimately, you just need to be able to model some tailing that is sufficient for 95% of all the features, and the rest you hopefully don't care about. So how is its feature finding now done in reality? I will not go into the details of the algorithms to a large degree. I will talk a bit more about that this afternoon when I'm talking about metabolomics, where this is done similarly. But roughly, what we do is we test for all possible charges. So you have to define which charge you're looking at. Usually charges between 2 and 4 or 5 are good enough. That's where you find most of the peptides. And for each of these charges, you determine what we call a seed list. So you say, where are the interesting regions? And an easiest way to seed your map is look at the highest peaks across these maps, because that's where the stuff happens. And you can work your way down. Once you have removed the highest peak, you go to the second highest peak. So there are ways to deal with that. Or you can actually seed by identification. That's another way. There's a specific tool in OpenMS for doing that as well, where you say, I only care about the things that I can assign names to afterwards. But then you also need to have some strategy of how you do that across different maps, because some of the stuff will be identified in one map, some stuff in the other map, so there are issues with that as well. And then for each of these seeds, we can look around the region, extend it, and collect all the peaks until we run into the noise, fit the model to the region, remove everything that's not consistent with the model, and call that a feature. That's a rough idea. Very high-level bird's eye view. In reality, we are using a slightly different strategy these days. We are looking at so-called mass traces. Again, something I'll explain more this afternoon. If you look at M over Z, we will see these peaks. These crosses here are peaks coming off at different M over Zs. And we are looking for series of peaks in adjacent spectra that are at roughly the same mass. That's what we call a mass trace. Now, question. If this here is a peptide, monoisotopic +1 +2, shouldn't these mass traces have the same length? Yeah, because if that is our mass spectrum here, then we should see all 4 peaks eluting at the same time. Could you speak up a bit? Yeah. These guys on the +1 +2, they tend to hit the noise earlier than the more intense peaks. Yeah, so that's what's happening. So what happens if you go to higher masses? It switches. So now the first mass trace will actually disappear earlier. But if you know the monoisotopic from the shape, you can still determine the upper boundary. You know, once you've figured out what is the most intense mass trace, you know that all the other ones that have lower intensities in your isotope model, they should disappear earlier. And that helps you to limit the range where you need to search for interesting peaks. Now, these mass traces, the examples I've shown you are kind of easy, but these mass traces can overlap in very naughty ways, and we need to be able to deal with that as well. Mass traces can contain more than one chromatographic peak. That means things are eluting in an overlapping fashion, but they can also be convoluted in other ways. So let me give you a couple examples. These are what we call interleaved features. Yeah, so you see there are 2 features, and the mass traces of one feature are between the mass traces of the other one. That is not a problem if you have high resolution and you can resolve these mass traces, but you can imagine if your resolution gets worse, then you start to see overlapping mass traces, and then it becomes very hard to disentangle. The second case is overlap, perfect overlap. So that would mean that the monoisotopic M over Z of both features is identical. Does that happen often? What are possible reasons for that? Depends on your column, yes, but then you would have to have a leucine and isoleucine variant of the same peptide in your sample, which again is not that likely. Note also they're very close in retention time. So they are deuterated? No. What would deuterated look like? That would also be shifted in this dimension. So this could be a doubly deuterated peptide. Whereas deuterated would also have a larger shift in retention time, usually. So this could be an example of carbon, heavy carbon labeling. But there are more biological processes lead to that. For example, cis-prolines. They tend to have similar retention times. Of course, they have the same mass. They're just isomers of the same thing. And they tend to introduce a very subtle shift. So that happens. And if you see that, then you can check that by looking at these mass traces. You basically do an additional peak finding on the mass traces. And then you need to decide how to split this guy. You can basically fit two peaks to that and pull them apart. And this overlap here is sort of the equivalent. We are shifted in mass to charge, but we are sitting on top of each other in retention time, in the retention time dimension. And then here the problem is really that you lose some intensity. This is going to be overestimated, and this is going to be underestimated in quantification. It's something that is not that easy to pull apart. Okay. The way we do that is by testing all these different models for different charge states, for different compositions, and we just decide which of these models fits best. So once we have decided on a seed or mass traces, we throw different hypotheses at the data and ask, well, does it look like a +1 or like a +2 or like a +3 feature? And they, as you can see, they would look distinct. And how do you test that? Well, we do a simple quality of fit model. We simply correlate the model at the position Ri of each peak Di. We normalize that. And if this number is better for one model, then we say this model fits better than the other. Very straightforward way to do that. In some cases, we cannot resolve these features unambiguously. So let's look at this guy. What do you think that is? That looks a bit odd. What's odd about this feature? So first of all, the elution profiles are fine. I mean, there's some tailing there, but that's probably good. But what's odd is the relative intensities of these isotope profiles, because this guy here seems to be even a bit higher than this guy. What's going on? It could be two overlapping features, but it's not. Does one particular element come to mind? That's not typically found in peptides. Sulfur. It's sulfur. It's cysteine or methionine that induces this weird shift. So what we actually do is we check not only for these different charge states, but we also check for the presence of sulfur because they change the averagenes as well. So on the whole, this type of algorithm works reasonably well. There are a couple issues. First of all, low resolution instruments might not yield good isotope patterns. That problem has more or less disappeared, except for very large proteins, where we see that again. Peptides can overlap, in particular, in complex samples. What do you think? How many features are there in a large run, proteomics run? Any idea? Well, let's try and do the math. If we really have 10,000 proteins in there, what's the average length of a protein? Well, guesses. Let's say 500 amino acids. That's rough, probably a good estimate. how long are these tryptic peptides? Well, we've seen the average is around 10 amino acids. So you can try to do the math. You get lots of peptides per protein that we see. And that means we usually see over 100,000 distinct features, because each of these peptides can also give rise to multiple features if you see +1, +3, +4 potentially. Most peptides, by the way, don't do that. They prefer one particular charge state. So peptides will overlap just because the gradient is not long enough to deal with that. This one solution could be to do fractionation. Then you actually split these 150,000 or so peptides across 10 fractions, and then you have to deal with less than that per fraction. Low intensity features also tend to be hard to distinguish from the noise. So you can tune these algorithms, and if you tell the algorithm to go down into the noise, what happens? Well, it's going to pick up all sorts of crap, and it's going to run for all eternity because it's trying to fit models here and there to all these tiny noise peaks. So if you see that your algorithm runtime goes up when you go to a new data set, then that's one thing to check, whether you actually have too much noise in this particular data set or inappropriately chosen parameters. And isotope labels, if you have them, that is something to keep an eye open for as well. If you do label-free quantification, you shouldn't have that, but we had cases where people did actually try to run it on isotope labeled stuff, and then all sorts of oddities can happen because the Averagine models simply don't fit that. You can correct them and tell them what they need to look for, but out of the box, that would not work. Let's talk a bit about alignment. There is a breadth of literature on how to align chromatographic runs. Dynamic time warping, linear, non-linear alignments. I've seen many PhDs spent just on trying to correct crappy chromatography. Personally, I would argue if you have to go to great lengths to correct the chromatography, then you should find someone who can actually do chromatography. But okay, some deviation will always be there. So how can we correct that? You can do that on the raw maps, but that means processing very large amounts of data, and it's very hard to sort of connect the dots on these maps. It's much easier to do that on the feature level when you have turned all these peaks into kind of landmarks. You know where these things are, and that makes the alignment problem a lot easier and also a lot faster. So rather than trying to figure out which of these 350,000 peaks here corresponds to which peak here in the original spectra, you do the feature finding, and suddenly you have only like 10,000 dots on the left-hand side and 10,000 dots on the right-hand side, and that makes it a lot easier. Of course, looking at these maps, if you would do that manually, it's not totally obvious that this dot here would correspond to the same dot on the second map. So what I'm going to show you is sort of the most basic algorithm for that. As I said, there are many different ways to do that, also non-linearly, also in OpenMS. But I just give you some idea of how this can be done in order to guide your thinking a little bit. What does this really do? Because the biggest effect is really a shift. And that is caused by either shift in temperature or problems with the injection, change in the dead volume. There are many things that can happen. But it's not common that sort of the retention time goes up and then down again and changes drastically over this whole map. It's usually a linear shift that is applied to the whole map. And that is why the simplest alignment algorithms are linear alignment algorithms, and they take care of, say, 90% of the shift, and the nonlinearity is the rest, and usually occurs at the beginning and the end of the map. So there's an algorithm by Lange et al. That tries to find pairs of features between maps and align them in a way that you minimize the distance between those two maps. So you can think of this algorithm as follows. We have a bunch of maps, map 1, 2 through K. And we will solve this problem in a pairwise fashion. Now, how do you, if you can solve this in a pairwise fashion, so go map 1 with map 2, how do you go from this pairwise problem to doing 50 maps? Well, the easiest way is to choose one map as a reference point and then do the pairwise thing for all them. So what you, in sequence analysis, what you would call star alignment. You align all pairs against the same reference. And if you do that, and if you do that properly, then you would end up with all these maps being aligned. And this is what we would call a consensus map, where everything has been laid on top of each other and where we link the features from all these maps into groups. And these we call consensus features. So how can we do this pairwise alignment? So the problem really is to find an affine transformation that takes the features from one map and minimizes the distance of these transformed features to the features in the second map. That's a very straightforward optimization problem. We can formulate that as an optimization problem, but there's a much faster way to do this. So what you can do is you can apply so-called post-clustering. Somehow part of the slide disappeared. I'm sorry about that. Fundamentally, you know that the mass over charge ratio is going to be fixed. A feature from here will not link to a feature that is 20 Dalton higher. The masses are fixed, so you need to look for corresponding features only in a very thin slice along the M over Z dimension. What you can then do is you look at all pairs and just throw arbitrary pairs, random pairs together and determine what would be the correct linear mapping for these two pairs. So what will happen is that you match up the right pairs, corresponding pairs, and you match up incorrect pairs. And now you look at the parameters A and B of your fitting, and lo and behold, the random matches will scatter all over, and only the correct matches will sort of give you the same values for A and B again and again. It's a very simple clustering algorithm that gives you, with runtimes that are below a second, gives you a very good approximation of a linear fit for that. You can refine that then, and that would be a nonlinear alignment step. And one way to do that is so-called lowest locally weighted polynomial regression. That takes the points that we have based on the linear fit and then tries to find a better approximation. So this paper here shows you an example where this lowest regression has been used. This here would be the linear alignment, and this here would be a lowest regression over these pairs. And you see, indeed, retention time is fitted better. So that looks great. Does that make a difference when you link these features? No. Why not? Because you don't have to get the retention time exactly right. In mass over charge dimension, things tend to be reasonably sparse. So whether you get the retention time aligned to plus minus 10 seconds or plus minus 5 seconds, the overall neighborhood will be reasonably dense. So this does improve things, but only in a small percent range. It's really not a major issue. Okay, feature linking is then the process of taking these aligned maps and assigning one feature in one map to a feature in another map. You can do that across maps for label-free quantification, or the same concept applies to labeled data. If you have two labels in the same map, you can cross-link within the map and find pairs of features that are, say, 8 Dalton apart for Silac, for example. In order to find matching pairs, you need to have a mass tolerance and you need to have retention time tolerance as input. And once you run that, you will get these consensus features. And in top view in the viewer, you will be able to see them as stars when you overlay multiple maps. It's going to show you the center of this map and all the other features from the other maps that are slightly misaligned to that. So in OpenMS, we have a bunch of tools that you can try to do that. The simple map aligner post clustering is what you can use for linear alignment, and then the feature linker unlabeled QT, don't ask about the QT, does a clustering and links these features and gives you back a consensus map. When you look at that in top view, then this is what you will see. So this is an overlay of several maps, and you see the consensus feature, this yellow dot. That is where these features are after alignment. And then you see these guys here. These are the locations of the features that form the consensus feature in the various maps. So you see for some of them, they're shifted to lower retention times. For some of them, they're shifted to higher retention times. You don't see much of a mass shift. Why is that? Because the mass security is much, much better than the retention time stability. But if you actually zoom in, so if you zoom into a thin slice, then these consensus features actually look like that. Because you also have a mass error, but you see the mass error is so small that here the numbers on the axis actually don't show it anymore. But fundamentally, you have the same type of deviation also in the mass to charge ratio. It's just very tiny compared to what you see across retention time. If you then want to check how well your data set behaves, you need to do some sort of quality control, and you can do various things. I'm not going into full-blown quality control here. I'll just give you some pointers of what you should observe if you have large data sets. So this plot, we call it the bathtub plot for obvious reasons, is something that scares many people. It's fundamentally straightforward. We have 20 samples. And what is plotted is the count of the features in the map, consensus features, that have a certain size. And by size, I mean occurs in n samples. So this bar here indicates that every feature in this consensus feature has been found in all 20 maps. This bar means these are the ones that are lacking in one of the maps. And these guys here are singletons. They have been found in one map and one map only. And why is that scary? Well, because it seems to imply that you can't really quantify across maps, because the vast majority of what you see will not be found in other maps. Is that a problem? There's an easy solution to that. Don't do replicates. Because if you don't do replicates, I mean, this plot will look just fine. Yeah? All the features have been found in all of the maps that you looked at. What happens if you don't do 20 replicates, but if you do only 3? Yeah, it will look much more sane. This feature detection means that there's always a probability of missing a feature, in particular for the low intensity features. The high intensity features are fine. They can be found across all maps. But most often, the interesting biology doesn't happen with the high intensity features. It happens in the low abundance stuff. And this low abundance stuff you will not be able to find in every single run. Now, if you have a 99% probability of finding these features, then you can compute the probability of finding things in all the features. And as your number of maps goes up, the probability of finding everything in all the maps goes down exponentially. So the more you do, the more you find these oddballs, but you shouldn't care about them, because there is still going to be enough signal in this part of this plot to give you years of follow-up. The second thing we need to do is some sort of map normalization. we discussed this strategy of internal standards already. That is one way to do it. It's not what I would recommend. There's a much easier way, depending on the type of samples. And we just rely on biology to solve our statistical problems. Sounds a bit strange, but what is the biology there? Well, it's cellular homeostasis. If cells are alive and if you compare similar things, then the vast majority of this cell is going to behave the same. You will have deregulated proteins, but all the housekeeping proteins, they will be more or less at stable concentrations. You will not see, even if you do heavy perturbations, you will not see 50% of the proteome being differential. It just doesn't happen. If that happens, then your experiment is probably not a meaningful one. So what we can easily do is we just assume the majority of these signals should be preserved. And even for analytical reasons, if you injected a bit less in one sample, then they should all be behaving the same way. So there's a simple way to do that. Robust regression. You just remove everything that looks odd and do that iteratively. It's a very simple thing. You just remove outliers that are too far away, recompute, and in this way you can recalibrate that. This here are two replicates, and this is before normalization and after normalization, and you see the distance between the three is much larger before normalization. Even with this simple robust regression, you can reduce the variance more easily. But there are, of course, better ways to do that. What we can do and will do here is integrate this feature finding into a NEU pipeline. And from this NEU pipeline, we can then do proper statistics downstream that I will not discuss here because that's going to happen in the MS Stats course. What do we need in order to make that happen? Well, a bunch of input files, label-free runs. We have a loop. We run a feature finder on that and the loop. And this is going to create a table. A feature table where we have for every row in the table one feature, and we have the intensity, a retention time, charge, and so on for this feature. And then we can use the map-aligner post-clustering to link them, and the feature linker unlabeled, and what comes out is a table that has features and has intensities across different runs in that. A simple matrix format. And we can take that, and there is a tool that's called MS Stats Converter. And MS Stats Converter takes the peptide intensities and some experimental design that you provide, and then generates an input file for MS Stats. And then you can use the downstream processing of MS Stats from this point, for example, to generate nice QC plots in a very convenient way. And you can embed that in NEU and get a processing workflow that includes OpenMS and MS Stats nodes in one go. All right, that's all I want to tell you. I think it's enough for now. That should keep you busy for a while, but I'd be happy to take a couple more questions if you want.